Polymers of carbohydrates. Okay, right, where do we begin? Well, let's start with the word that we're going to be using a lot during this course, polysaccharide. Poly for many, double C in saccharide. What is a polysaccharide? Well, it's many monosaccharides joined together. How are they joined together? Well, they're joined by condensation reactions. That's the type of reaction, remember. Again, we can say saccharide, anything with a saccharide in it is a carbohydrate, is a sugar. Also OSE, O-S-E, hexose, glucose, dextrose, any of those things are sugars. The polymers, there's 4 that we need to know the names of. We have cellulose. Again, we've got our OSE. Yep. Now, there's 2 that live within the same family. We've got starch, and starch is made from 2 things, amylose and amylopectin. And the last one is glycogen. So we're going to do these in 4 columns. Okay, we're going to look at where do they exist? Well, first of all, we've got plants. Plant cell wall. These 2 are both in plants. They are glucose or / energy storage. And we've got animals, energy glucose. I'm going to put NRG because I'm short on space. Energy, storage. NRG, energy. It's a shorthand if you're short on space. I use a lot of shorthands. If you are going to be using things like this, I used concentration with a little n in it, a superscript n. If you are going to be using abbreviations, I use up arrows to mean increase, down arrows to mean decrease. These are all fine. I encourage it. It makes sense. It makes your notes really quick to make, and it's more efficient in total, and I advocate that at all times. However, make sure that you're consistent. You use the same abbreviations the whole time, because if you use it once or twice and then forget it, you go back to your notes, you don't know what the hell it means. What is it made from? Well, cellulose is a polymer of beta-glucose, and only beta-glucose that you're going to find in cellulose. The others are all alpha-glucose. Okay, what type of bonds do we have? Well, they're all carbohydrates. They all have glycosidic bonds. Now, I'd recommend writing this out every time. I know it's boring. You could bracket them together, but you need to get it grained into your head. It's a carbohydrate, glycosidic, 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 every time glycosidic. Boring repetition, I know, but that repetition is going to help that sink into your brain. Okay, now a bit more about the structure. Let's do these one at a time now. So cellulose is unbranched, or you could say straight chain. Now, remember we've got glucose is a hexagon. Don't forget the oxygen. We've got our hexagon like this. In the middle is our glycosidic bond. Now, beta-glucose. Remember beta-glucose? See if you can think about the structure. What's different between beta-glucose and alpha-glucose? If I've got space, I'll draw them at the bottom here afterwards. But with beta-glucose, the OHs, one is on the top and one is on the bottom. Remember Santa. Alpha-glucose, ho, ho, ho. The OHs are both on the bottom in alpha-glucose. In beta-glucose, they flip over. And that's why the oxygen flips over on both sides. So I'm going to draw a few of our... Don't forget our little extra hydrogen there. Carbon and hydrogen. So this is our straight chain. Now, we could draw this straight chain. Obviously, we don't have to draw all the hexagons. And I'm going to draw, so we're just going to draw a straight line. And then we have lots of these straight lines. I'm going to draw 3. In reality, there's going to be more than that. And between these straight lines, you had another row of green hexagons up here. We get some positive forces and some negative forces going on, and they create, have a think, see what you can guess what they might be. They create hydrogen bonds. So these, in fact, I'm going to change color to label it, it'll be easier to see. These are called microfibrils. These long straight chains of beta-glucose. And they are joined together, or held together, by hydrogen bonds. So how does the structure of glucose affect its function? Well, it's very strong, which means it provides structure for plant cell walls. It is also insoluble, which means it doesn't dissolve when water is around, which is quite useful if you're going to be a structural compound, especially in an aqueous environment. Okay, so moving on. So now we can look at amylose. Now, the function of these two, both in plants, they're both storing glucose, which are going to be respired. In fact, I might even put the reason, obviously, we want glucose is for respiration. We've both got glycosidic bonds. They've got a different structure, though. So here, I'm going to draw, in fact, let's divide all of these. It's about as straight as my writing goes. Obviously, they have a shared function. This is also unbranched, but it's also and coiled. This means it is compact storage of glucose. So it doesn't take up too much space. Pretty efficient, dense energy store. Now, its structure unbranched and coiled, whereas this is unbranched and straight. This is to do with our properties of alpha glucose. So I've drawn that on a bit of a wonk already. But what happens is the angle, and this is alpha glucose, so the oxygen is going to be in the same place each time. We end up with this non-straight angle. So if I were to draw that, it's going to start like this, and it's going to coil, if we drew a much longer molecule. Now, this is something we can test for, and I'm going to explain about the test just at the end. It's very simple, a test for starch. It is also insoluble. Now, the reason that this is important here is much more important. It doesn't affect the water potential. See if you can remember what the symbol for water potential is. It's this Greek letter, looks like this. And the importance of this has no effect on osmosis. That's a key term, osmosis. I should probably have written that in red. This is really important. If this was soluble, if we just stored our glucose as glucose, which is soluble, inside the cell, it would have a very, very negative water potential. So there'd be lots of pressure for the water to rush in by osmosis. And if the water rushed into every cell by osmosis, you're going to strain, or it's going to start basically be pumped full of water and then possibly burst, depending on how strong your cellulose is. Now, amylopectin. Same compound, alpha glucose, glycosidic bonds. It is, however, branched, which the reason that this is useful is increases the surface area. And why is that important? It can be hydrolyzed more quickly. So if you imagine trying to get the glucoses off these, you can basically only break off one glucose at a time because it's attached to each one. If you've got branches in here, then it becomes much easier to get our glucose out. In fact, what I haven't done on each of these, I've forgotten, as I told you not to do, is to make sure that we have our extra carbon atom, otherwise it's only got 5. Okay, so again we've got a bit of an angle, it's alpha glucose, so that it stays in the same place each time. But what we would have is another one coming off here. And so we have our branch. And this would continue happening, you could get branches off here. So again, if I was to draw this in our simple line version, something like that. It is also insoluble, has no effect on, this time I'm just going to use the symbol, and therefore no effect on osmosis. Now this takes us to glycogen. I've left this column a little bit narrower. Sorry if you're copying this live, then, I mean, it shouldn't make too much difference. There's just a little bit less writing. If you fitted all this writing in, it's not going to make too much difference. The only difference between amylopectin and glycogen, there's two. In fact, I'm lying to you. Amylopectin is glucose energy storage in plants. Glycogen is energy storage in animals. Now, what do animals do that plants don't do? They move. They move, which means they need to respire more quickly. So if they need to respire more quickly, what are they going to have? Their compound is going to be, it's almost the same, but it's going to be very branched. It needs a bigger surface area, and the bigger the surface area, the faster you can break down the glucose, the more you can then respire, and the more ATP you can make. So I'm going to put here, very branched. In the exam, branched will get you the answer, but I'm going to say bigger surface area, faster hydrolysis. That's probably all we need to know. I'm not going to draw the diagram like this. I don't think it's necessary. But you get the idea. Something like this. All these diagrams that I'm drawing in general in biology are a horrendous representation. They don't actually look anything like this, but they are resembling the important factors. And there's also insoluble, no effect on the water potential or on osmosis. Now, I kind of broke one of my rules here. I'm actually going to underline osmosis because this is very important in this question. It very often comes up. So that is all of the polymers of carbohydrates on one page. So starch, amylose, and amylopectin is made. It's a compound of these two. Oh, I haven't done the test for starch. Well, do I have space at the bottom here? In fact, maybe I can do it at the top. Well, the test I'm going to tell you very quickly. You add iodine. If starch is present, it turns blue-black. So, test 1. Add iodine. You can say potassium iodide. That's absolutely fine. That's technically what the compound is. But in the exam, they accept iodine. Add iodine. It turns blue, dark blue, or you could say blue, black. It's very dark, depending on how much starch is in there. If it's negative, then it stays orange. I don't have an orange, so it's a bit of a rush. I'm sorry, but I'll explain that again. You've got a mystery substance. You want to find out whether there's any starch or not in there. You add some iodine, which is orange in color, and then if it turns blue or black, then you are certain that there's some starch in there.
To get more biology videos from me, subscribe to the channel, and of course don't forget to try out my complete A-level course in the free trial over on the Tailored Tutors website. Up next, you can transform your grade in about an hour with my grade transformations playlist, or you can watch the video that's been magically selected for you from the people at YouTube.